Hello, good afternoon, everyone. As I promised last week with my first video on Ujian Amali Sains last week, I did a question on Form 4 topics. So today is 1 week from last week, yeah. So I'm trying to do a Form 5 topic today. Okay, so this question has not come up before. Okay, in fact, not many questions have come up before because this is only the third year of Ujian Amali Sains. So I'm sure most of you already know that the first year we had it in 2021, and the question that came up was vitamin C. And then the second question, last year was the second year, and they had this anaerobic respiration, which is fermentation right, by yeast. So today I will try to get a question. Actually, I've done a question on Form 5, which has never come up before. It's not your usual question because you don't have the MV, RV, and CV, that kind of thing. But it is a question that is worth doing, that it is possible that it can come out, okay, in any experiment. It's actually quite simple. So do not worry about trying to think of very complicated questions. Actually, this Ujian Amali Sains, just think that within 40 minutes, what are the experiments that can be done? So you'll find that not all experiments can be done within 40 minutes. Okay, so you will, by that way, you can already narrow down what are the experiments or what are the questions that cannot come up. Okay, so without further ado, let me go through today's question. So I'm sure many of you will not expect this question, but it's a very simple question to do. Okay, if you know your your theory well, you will be able to answer. Don't have to worry so much about your Ujian Amali Sains. After all, it's only 15 marks. Okay, come, let's try. Let's start this today. All right, what is the question? All right, now you can see a map here. So there's something to do with a map. All right, so who is here with me today? Okay, I don't see anyone yet. So it's all right, we'll just continue, yeah? Okay, so what is today's question? All right. Today's question is on, oh, sorry, I opened something else. All right, now, quadrat sampling. Okay, this is a topic is a question in chapter 9. This one, dynamic ecosystem, right? So quadrat sampling is a technique that can be used to find out the distribution, density, or estimated population of an organism in a habitat. Okay, now don't worry, you don't need to go to the field, right? This will just be a simulated experiment where you can do it in the lab, all right, where you give you a map with a map that represents certain species on an island or on a field or whatever, and all you have to do is to create a quadrat and try to find the population, or what you call either population or density or whatever the question wants. Okay, there are only 3 formulas. You have to check back your chapter 9.3, I think, population ecology. There are 3 things you can find out from here with this quadrat sampling. This for plants, yeah? Don't forget that it's only for plants, not for animals. Okay, now what are the 3 things you can find out from this with this quadrat sampling? Number 1 is density. How densely populated are these trees or these plants in that area? That's 1. Number 2 is the coverage, percentage coverage. Okay, that means these are usually for grass or moss. Okay, because they do not have one specific tree that you can or plant. So you find that it's like coverage. And the third one is frequency. Okay, so let's go to the question. In this experiment, you're required to study the distribution of plants in a given area. You're given a map of an island which shows the distribution of 2 different species of plant. Okay, I'll show you the map afterwards. Let's look at the procedure. Okay, procedure. First one, using a ruler and a pencil. So you got to make your own quadrat. Okay, a quadrat will not be 1 by 1 meter. That'll be too big, yeah? 1 by 1 meter is for you to actually bring it into the field, right? To put it onto the grass and find your calculate the number of species that appears in the quadrat. But here, because you're doing on a piece of paper, So you'll be asked to draw a quadrat, all right? And then follow the instructions. Here says draw a square, a perfect square, yeah? Measuring 3 cm by 3 cm. So this is going to represent a quadrat. And using a pair of scissors, cut out the cardboard. So you have a small little square like that, all right? And use this square to represent your quadrat. Okay, this is going to be a quadrat. And you're going to put this, I'll explain in BM, yeah? Okay. All right, I'll try to do in BM. In fact, I can, but it'll take a longer time, right? Because we'll double the time. Okay, so quadrat sampling, ataupun persampalan quadrat ialah teknik yang digunakan untuk mencari density, iaitu ialah kepadatan, okay, dan juga populasi sesuatu organisma di dalam habitat. Okay, dalam eksperimen ini anda diperlukan membuat satu eksperimen. Anda diberikan satu peta yang melambangkan satu pulau yang mempunyai dua jenis pokok yang berbeza. Jadi dengan menggunakan prosedurnya, menggunakan ruler iaitu pensel dan juga ruler pembaris dan juga pensel, lukis satu segi empat tepat, ya. Segi empat, okay, square, perfect square, berukuran 3 cm dengan 3 cm atas cardboard, ya. Anda akan dibekalkan cardboard dan menggunakan cardboard, menggunakan gunting, potong cardboard itu. Lepas itu gunakanlah cardboard ini sebagai quadrat. Okay, now the rest of you I think you can read in English, right? So I'll explain in BM lah. So letakkan cardboard ini sebagai quadrat anda di mana-mana bahagian map. Pada peta tu letak di mana-mana sahaja, tidak terikat dengan apa-apa syarat. Letak sahaja di mana-mana dan lukiskan outline, iaitu garis di luar. Okay, so put your square, draw the outline, iaitu garisan di luar. Again, you can take away the cardboard. Anda boleh angkatlah cardboard itu. Okay, sekarang ulangi langkah yang kedua ini. Please repeat, do again. Dan lukiskan 4 lagi kuadrat. Draw another 4 more on the map. So make sure, pastikan semua kuadrat ini masuk di dalam pulau itu. Okay, on the map. After saya show the map. Dan labelkan sebagai kuadrat 1 hingga kuadrat 5. So you're asked to make 5 quadrats. Anda gunakan kuadrat ini, segi 4 kad ni, untuk membuat 4 kuadrat, lukiskan di atas 5 kuadrat di atas map. Okay. Lepas tu, dalam setiap kuadrat ini, kira ada berapa pokok A dan berapa pokok B. Actually, my lesson here is on in actually supposed to be English lah, right? But if you want in BM, just let me know. Saya akan I will explain in BM lah, right? Okay, Carrie, good afternoon. Yes, thank you very much for joining. So the next thing you need to do is, lepas kira, anda perlu buatlah satu jadual untuk mempersembahkan data anda. Okay, so simple, sangat senang. Okay. Jangan lupa apa-apa saja eksperimen yang keluar, ia pasti boleh dijalankan dalam 40 minit. So jangan bimbang sangat. Eksperimen yang susah-susah, yang memerlukan banyak pemasangan, okay, biasanya tak akan keluar. Mostly it will be a simple experiment. The difficult part is the question, which you will never know. You cannot expect because it will not be the same as your textbook. Soalan itu tak akan sama 100% seperti dalam buku teks. Dia akan ubah, dia akan olah, they will change the question. But if your facts, this one is very, very good, very solid, anda akan dapat jawab. Okay, next. So katakan ini ialah peta yang anda dibekalkan. Anda bekalkan. Okay, dibekalkan. So titik-titik ini melambangkan pokok. Okay, plan A yang berwarna merah, plan B yang berwarna biru. Okay, so soalan, ya, anda boleh jawab soalan ini sebelum anda lukis kuadrat itu. Soalan pertama, dengan melihat saja, looking at the map, okay, which species yang manakah adalah spesies dominant di dalam kawasan ini? What is the dominant species? Okay, berikan reason, ialah penjelasan untuk menyokong jawapan anda. Okay, why you say is A or why you say is B. Okay, anyone to answer this? Boleh anda jawab ini? Siapa? Nama okey tu. Okay, nama okey tu boleh jawab. Apakah spesies yang dominant? species dominant di dalam kawasan ini? Okay, what is the dominant species? Carries, or maybe carries you can answer. What is the dominant species? It's very simple to see. Anyone wants to try? Okay, I give you maybe another 10 more seconds. Okay, if not, then I will give you the answer lah. It's very simple, either A or B, right? There's no other answer. Okay, so anybody? All right, no, yeah. Okay, so the answer is it's B, ya. Plant B, tumbuhan B. Okay, kenapa? Why tumbuhan B? Because you see that, look at the bilangan, the number, right? The number, the one that is more, okay, the Ama, correct, plant B, because the number of plant B, a B plant is the most in this area. Which one is most? The most will be the dominant species. They will grow very well. They might even, have inter-specific competition with other plants because this plant is well adapted. Adaptasi plant tumbuhan ini sangat baik terhadap kawasan itu, di kawasan itu. Jadi dia akan tumbuh dengan subur, tumbuh dengan baik, dan dia akan membiak dengan banyak. Jadi dia akan menjadi tumbuhan yang dominant, the dominant species. So they will reproduce, right? Because they are very suited to stay in the area. Okay, now state 2A biotic factor. Nyatakan dua faktor abiotic yang akan mempengaruhi distribution is called taburan populasi tumbuhan di dalam kawasan ini. Taburan maksudnya berapa banyak, adakah di tempat sini sahaja yang ada, ataupun dia tumbuh dengan banyak ataupun tumbuh dengan sedikit. So what are the factors that can affect whether the plant grows a lot or grows very little? Apakah faktor yang akan mempengaruhi? Anybody to answer this? Abiotic. If you learn in chapter 9, ada faktor biotik dan faktor abiotik. Faktor biotik ialah faktor benda-benda yang ada nyawa, benda yang ada is alive. For example, animals, plants, bacteria, itu ialah biotic factor. Kalau abiotic factor, faktor-faktor yang ada di situ is not alive, like the sunshine, the air, the amount of carbon dioxide, the acidity of the soil, okay, tekanan udara, okay, semua itu adalah faktor yang bukan hidup. Okay, jadi faktor bukan hidup ialah faktor abiotic. Jadi maksudnya, okay, tak ada jawapan lagi. So abiotic factors could be light intensity, keamatan cahaya, pH of the soil, pH tanah, temperature, suhu, water, air. Okay, berapa banyak air yang didapat di kawasan itu. So many jawapan, ada banyak jawapan tentang abiotic factor. Okay, you can refer to your textbook. These are only 4 examples. Okay, let's say you have done the procedure just now. So it should look like this. Katakan, anda letak di mana-mana sahaja. Okay, make sure you draw 5. Alright, now everybody's answer will be different. Okay, your answer and your friend's answer, you will not be drawing on the same place. It doesn't matter. When the paper, dia akan lihat jawapan anda sahaja. Okay, bila anda lukis di situ, pastikan anda lukis dengan jelas. Make sure it's clear, and then you do your calculation there. You do your calculation there, and then you will check the calculation whether it's correct or not. Okay, so based on the distribution of the quadrats on the map, okay, berdasarkan cara anda letak kuadrat di atas peta tu, apakah jenis sampling ataupun persampelan yang anda telah lakukan? What is the type of sampling that you have done? Okay, what is the type of sampling? Okay, good day. pH value, yes, Danish Vari, correct. pH value is the abiotic factor. Yolekin or Chegu. Okay, it's a Chegu, yeah. Okay, thank you for joining the class. Yeah, plant A, carries, it will be not plant A, yeah. Plant B is the one that is the dominant, because you see, you look at the number of plants here, the blue dots are more than the red dots. Okay, blue ones, you just take a rough, quickly just count up. You can see clearly that the blue has got more compared to the red. Okay, you're not sure, just make sure you count first. Okay, what's the type of sampling? Ada dua jenis sampling. How you letak, macam mana anda letak kuadrat itu, ada dua cara. Satu cara ialah anda buat secara rawak. Rawak means random. You simply letak mana-mana anda suka. You just put anywhere you want. There's no pattern. Like you can see here, this is random. Okay, because the question told you just now, just put anywhere you want. There's no specific place. Mesti letak di penjuru atas. Lepas itu, then you calculate 1 cm away, put another one. Another cm away, put another one. Kalau dia ada jarak-jarak tertentu, okay, how many cm, you put one, and then another cm, put another one. That is not rawak. Itu bukan random. Itu ialah systematic sampling ataupun persampling systematic. So here you can see that this is random sampling. Okay, so this is what you need lah. Either random sampling or systematic sampling. Systematic sampling means there is a certain distance for every quadrat. You put one already, then you measure a certain distance, then you put another one. After that, you measure a certain distance, you put another one. Okay, now let's look at your table. The third one, yeah. In the space provided, construct a table to show your results. All right. So to show your results, make sure you have all the data that's needed lah. You got to have your number of quadrats. Okay. So look like this. Quadrat 1, 2, 3, 4, 5. Because you make 5 quadrat, anda telah ambil 5 persampalan quadrat. Dan setiap satu tu anda dapati ada A dan B. Kalau tak ada, letak kosong. Macam, look at this one. Quadrat 3. In quadrat 3, you don't have A. You only have B, right? So put 0 there. And then 2 for the B. B species is the blue one. Alright, so you just put whatever number here and then you total it up. A ada jumlahnya 1, 2, 3, 4, 5. B, you have 3, 1 plus 2 plus 2 plus 3 is 11. Okay, so this is the total number of pokok ataupun tumbuhan that you find in your quadrat. Alright, so like I said, everybody will have a different answer. But it doesn't matter, we want to know your calculation. Most importantly is your calculation, whether you did correctly or not. Okay, then next question. After doing this, now this is 2 marks, yeah? So usually table is either 2 or 3 marks. Alright, next. Given the area of the island is 50 meters square, let's say this map represents an island and the area is 50 meters square. And the area of 1 quadrat, the area, luas 1 quadrat is 0.5 meter square. So 0.5 meter persegi. Find the density. Okay, density, there is a formula. Usually the formula will be given to you. But if do not, just in case they don't give to you, try to memorize that. It's not that difficult to memorize. Okay, density, they ask you to find density. That means how close are the plant in that population. Like it's a, what they call purata. Purata bilangan pokok per meter persegi. In 1 meter square, how many plants do you have? Okay, so formula, remember the formula first. Density equals the total number of plants. Okay, kepadatan. Density is kepadatan. Bersamaan bilangan jumlah semua pokok yang dijumpai dalam semua kuadrat. Okay, so divided by, dibahagi dengan bilangan kuadrat. How many quadrat do you use? Anda gunakan berapa kuadrat? And also times the area of a quadrat. Apakah luas 1 kuadrat itu? Okay, so you look here, lihat di sini. Total number of quadrat in all, total number of plants in all the quadrat is this one. So you have to take, you're looking for A, yeah? Plant A, yeah? Okay, let me use my pen here. Plant A. So you're doing plant A, right? So your plant A, the total is 5. Alright? And then the number of quadrat is also 5. You use all together 5 quadrat. And the area of 1 quadrat is 0.5 meter square. So your pengiraan atau the working 5 bahagi, then at the bottom is 5 quadrat times 0.5. Okay, so that means 5 bahagi 2.5. Jawapannya ialah 2. Okay, apa unitnya? Unit ialah per meter persegi. Because di bawah ini ialah area. So your unit is, it means in 1 meter square, roughly there will be 2 plants there. The density ialah 2 pokok dalam 1 meter persegi. Okay, 2 trees or 2 individuals in 1 meter square of the area. Okay, so you remember the formula lah. You add up all the number of plants that you get inside your quadrat, divide by the number of quadrat anda gunakan berapa, anda gunakan 5, dan apakah they will give you, what is the area of the quadrat they have to give you. Kalau tidak, anda tidak boleh buat. Okay, for example, just now we said 3 by 3, isn't it? 3 by 3 is representation. Alright, 3 by 3 is a representation. This is just a representation only, but they want to give you this one, so you use this in your calculation. Okay, because if you put 0.5, your quadrat is too big, you can't put it on your map. That's why you put 3 by 3, just for simulation. Simulasi sahaja. Okay, next question. Soalan seterusnya. Based on your data, okay, lagi satu pengiraan anda boleh buat. That is, dia boleh minta anda buat cari frekuensi. Okay, frekuensi ialah kekerapan. How often do you get this plant dalam kuadrat anda? Berapa kerap kuadrat ini ataupun tumbuhan ini akan ditemui dalam kuadrat itu? Okay, apakah formulanya? But this is for B untuk spesies B. Jadi anda tengok spesies B Alright. So formula for frequency is number of quadrats with spesies B. Tengok ada berapa kuadrat yang ada spesies B. So you see 5 quadrat, 1, 2, 3, 4, 5. All the 5 also got at least 1 pokok B. Okay, all the 5. And then divided by the total. Total is 5 quadrat. They are all together 1 quadrat, 2 quadrat, 3, 4, 5 with the species B. And then don't forget to times 100% because frequency is in percentage. Okay, so 5 times 100% divided by 5. This 5 means not this 5. Alright, this 5 is not this 5. This 5 here means I got 1, 2, 3, 4. All the 5 quadrat also ada tumbuhan B. Now, if there's no tumbuhan B, there will be a kosong here, isn't it? Let's say this is kosong. Let's say quadrat number 3. Quadrat number 3 don't have the plant B. It will be kosong, right? In that case, it will be 4. 4 out of 5. But now it's 5 out of 5 because all the quadrat also have tumbuhan B. Okay, so this 5 is this 5. Where is it? Sorry, here. This 5. 1, 2, 3, 4, 5. Not this 5. And this 5 here represents there are 5 quadrat. Altogether, you use 5 quadrat. Okay, so the answer is 100%. That means in all your quadrat, there will be at least 1 pokok B found inside the what you call inside your quadrat. Okay, so this is another calculation you can use or they can ask you to find. Frequency and density for plants. Alright, so this is for quadrat sampling is for plants, not for animals. Okay, next Suggest a method. Okay, suggest a method on how this technique, the technique used in this study, that means the persampling quadrat or quadrat sampling, can be improved. How can you improve this method so that you can obtain a more accurate result? How do you make it more accurate? Okay, now in statistics, when you learn about the statistics, if you want to make your data or your result more accurate, what we normally do is, kalau nak jadikan lebih tepat, lebih jitu, jawapan lebih tepat kepada bilangan pokok sebenar. Alright, we want to have a more accurate answer. What we normally do is we increase the samples. Means, maksudnya di sini anda tambahkan bilangan kuadrat. Here you only have 5. Now, if you are rajin, right? If you are hardworking, you got extra time. You don't do 5 only. You do 10. If you do 10, kalau anda ambil 10 kuadrat atau gunakan persampling, anda ambil 10 sampel, you will find that your answer after calculation will be lebih mirip atau lebih dekat kepada jawapan sebenar. Okay, so for this question, if they ask you how to make it more accurate, you always answer like this. You say that you increase the number of quadrat samples. That means instead of doing 5 is of course shorter time, all right, and it's easier to do lah. But your disadvantage is dia kurang tepat lah. Right? Dia punya jawapan tu mungkin bukan sebenar-benarnya, not so real, not so close to the actual answer. But if you have time, you do 10. Okay, jangan letak 5 saja, letak 10. Bertinggi pun tak apa, you draw another one on top. Alright, you do 10 times. You will find the answer will be more accurate to the actual number, actual data. I mean the actual result. Okay, state a disadvantage. Disadvantage of improving it by adding the number of quadrat samples. What's the disadvantage? Apakah kekurangannya jika anda tambahkan bilangan persampulan kuadrat? Of course, kekurangannya. Okay, anyone to answer? I think today everyone just want to watch me. Okay, good day everyone. Yes, nobody answer. Okay, so if you want to, the disadvantage is if you add too many, of course it will be very time consuming. Increasing too many quadrat samples will be very time consuming and also effort consuming. Okay, menambahkan atau menambahkan terlalu banyak persampulan kuadrat akan mengambil terlalu banyak masa. Okay, sometimes you cannot afford to go through the whole island and taking hundreds of this quadrat sampling, depending on how much accuracy you want lah. Kalau anda betul-betul perlukan data yang lebih tepat, anda tambahlah bilangan kuadrat. But sometimes you cannot do too many, so you don't have time. Okay, so this is the disadvantage of increasing the number of quadrat sampling. Okay, next. Now, why is this method or technique not suitable to be used or to estimate the number of population of snails on the island? Let's say, of course, there'll be animals there, all right? Small little animals in the grassland or whatever. Why is it not suitable? Anyone can answer this? I think this one you will know. This is simple. Anybody to answer why it's not suitable to use, tidak sesuai digunakan untuk mengira populasi siput babi di atas pulau ini. Okay, kenapa tidak sesuai? Anybody? Danishwari or Caris or the Amar or okay? Okay, no answer ya. Okay, the reason is because one difference between plants and these snails is perbezaan ialah snails boleh bergerak. Snails can move. So what is the problem if you use quadrat sampling for animals that can move? What's the problem there? The problem is if you put your quadrat there, right? Sometimes you randomly throw the quadrat. You throw the quadrat, it lands there. Maybe there on the spot there and then ada siput babi di dalam. Tapi bila you nak kutip, you nak kira, the siput babi boleh bergerak. Okay? By the time you walk over there and then you go and count, kira, the siput babi may be moving out of the quadrat. So, haiwan yang bergerak, haiwan yang boleh bergerak biasanya tidak sesuai. Not suitable for us to use quadrat sampling to find the population density. Okay, so it's not suitable because snail is an animal that can move. In fact, for ants also, right? Ants you put the quadrat there. By the time you want to go and kira, they'll be moving all over the place. You don't have an accurate number. Okay, in that case, jadi, okay, altogether there are 15 questions. We're coming to the last question already. Suggest a technique that is suitable to be used to estimate the population of snails on an island. If you cannot use quadrat sampling, what is the method that you can use to estimate anggaran populasi siput babi atas di pulau ini? Anybody? You sure know if there's a chapter 9 here you've learned already. There are two ways of getting. One is for pokok. Pokok is this quadrat sampling. For animals, you don't use this. You use another method. What is the other method? Anybody to answer? No answer yet. Nobody dares to answer today. Okay, the other one is what we call CMRR. Okay, if you do not know what I'm talking about, please go and do your revision. Because if it doesn't come out in your Ujian Amali Sains, this can very well come out in your structure. This can also be a question in the structure. Okay, why, if you don't use this for, what is the method that we use to measure the population of animals that can move? We use CMRR. What is CMRR? Capture, mark, release, and recapture. Okay, you put a trap, capture the animal at the first capture, then you mark it with a certain permanent ink, ink that is not washable by rain or water. Then you let it go, release. Let them mingle with other animals in the habitat that is not marked. And then you set another trap a few days later, and you recapture again. See how many of them are marked and how many are unmarked. Then put all these numbers in a formula. You will find the estimation of the population of your seafood body. All right, okay. So I see there are no questions here. Today is very simple. Maybe you think that this experiment is too simple. All right. Again, I don't expect experiments to be very difficult. It will be easy to be conducted. Make sure the decider is whether it can be done in 40 minutes. And if your theory is strong, no problem at all. One more thing, when you count this quadrant, if it's half, more than half you count. Okay, look, this one here, there's more than half you count. If it's less than half, it's out of the boundary of your quadrat. Kurang dari separuh jangan kira. Lebih dari separuh kira. Kurang ataupun sama, equally separuh anda boleh kira. Like this quadrat number 5 here. Okay, this one we don't count. Yang ini, we don't count. Okay, this one, where is it? This one we don't count. Okay, understand or not? Alright, okay, so this is all I have for you today. Very short, very sweet, right? Very short and easy to understand. If it doesn't come out in Ujian Amali Sains, it can very well come out in your question. Okay, so see, I have time, not next week, I may do another experiment to guide you before. I have only one more chance. Next week is your last Saturday before your Ujian Amali Sains. So depends if I have the time or not. If I have the time, I will be able to do another one. If not, then I wish you all the best for your Ujian Amali Sains. And don't forget, I've had previous videos about all the technique and jawab in in these previous videos on Ujian Amali Sains. I've already explained what kind of questions that can come out and how to answer the certain questions, hypotheses, MV, RV, and so on. Okay, so check it out if you haven't checked it out. Okay, so see you everyone. Hope to see you another time. Right, bye-bye everyone, and good luck in your exam. Okay, thank you. Bye-bye. I will give you my Telegram name. If you would like to contact me to ask questions, most welcome. All right, most welcome for you to ask me. Okay, bye-bye everyone. See you next week. Have a good weekend.